Good morning, gentlemen. Today is Thursday, April the 23rd, and today we are going to talk about section 16.2, how many proteins work together in order to replicate DNA. Now, the one thing I want to point out to you, and the one word that your textbook doesn't use very often, is that most of these words, proteins that we're talking about here, are actually enzymes. So they're proteins, but they are enzymes that help to replicate DNA. So let's get started. Oh, by the way, before I do get started, I'm going to break this up into three parts because this is a long section and there's a lot of complex stuff here. So first, the relationship between structure and function is it really comes to light when you look at the double helix. And it turns out that Watson and Crick's model with specific base pairs, with adenine going with thymine all the time and cytosine going with guanine all the time, suggested to them, Watson and Crick, that this might be part of the mechanism that was used for copying DNA during mitosis and during meiosis. So the basic principle is that the base pairs need to come together on a template of some sort. Okay, and so since the two strands of DNA are complementary, A with T, C with G, and so on, each strand, the two strands that are together, can be taken apart, and each can act as a template for building a new strand when the replication process takes place. And so what has to happen here is that the original molecule has to unwind, and then two new strands need to be built using the base pair rules, A with T, C with G, along those two original strands or the two sides as the templates. So it would look something like this. You start out with the original DNA molecule, which is labeled here as the parental molecule. And the first thing that needs to happen is we need to unwind it and separate the two strands. Okay, and that's what you see here. And so now we've got the two parental strands separated into templates. Now, by some mechanism that we'll talk about in a minute, free nucleotides need to be added to each of those strands, like you see here. And now what you've got in this and in this are two identical strands, identical to the original parent strand. So Watson and Crick used this model, and it's referred to as the semi-conservative model of DNA replication, because what it predicts is that each of the two daughter molecules will be half of the parental molecule plus new nucleotides. All right, in other words, one old strand derived or conserved, and one newly made strand. The other two models were the conservative model, where the original parent molecule gets back together and the new strand of DNA is completely new. And then what's called the dispersive molecule, where each strand breaks up into a lot of little pieces and is copied that way. So what that indicates, or what that would show here, in the conservative model, you would start out with the parent cell, the parent strand of DNA, and after, whoopsie daisy, one DNA replication, here's the original strand again, and the new strand is made of completely new nucleotides. And after the second strand, you'd end up with, again, the parental strand and three new nucleotides. In the semi-conservative model from Watson and Crick, the parental strand unwinds, and you can see here that each of the two new strands is half parental and half new. And then after the second replication, the original parentals are here and here. And in the third model, the dispersive model, the original DNA molecule breaks up into lots of little pieces and is spread out throughout the entirety of the four new strands in the end. It turns out that Watson and Crick were correct. All right, and experiments by Meselson and Stahl indicated this. What they did was they took bacteria and they cultured them in a medium, a broth, with heavy nitrogen. All right, this is N15 that you see here. Then they put them into another medium. I hate this mouse. With a lighter nitrogen isotope of N14. Now, the DNA was then centrifuged, and we ended up with um, uh, oh dear Lord, every time I touch the mouse, not even moving it, just touching it. In any event, the DNA is sampled after the first replication and after the second replication. And so what this indicates is that in the after the first replication, every single strand of DNA had the same mass. In other words, there was a mixture of the heavy and the light nitrogen in there. But after the second replication, there were two bands in the test tube, a more dense band with the heavy nitrogen and a less dense band with the lighter nitrogen. So what would the models predict? Well, the conservative model would predict that there would be two bands after the first replication, one that was made of completely new DNA and one that was made of the original parental strands. That doesn't work, all right? In the semi-conservative model, you'd expect one band, all right, because both of the new strands would be half old and half new. And then at the end, after the second replication, you'd expect two bands, one light and one heavier. Finally, in the dispersive model, after the first replication, you would expect one band, right? Each of the two new strands is 50-50. And then after the second replication, you'd expect, again, one band, all right? Because all of the parental DNA would be evenly distributed amongst the rest. And this doesn't work, all right? And so you can see that the semi-conservative model is definitely the one that works. So Watson and Crick were correct. Now, DNA is copied remarkably quickly, all right? That's something like 500 base pairs a minute in a typical bacteria cell. A little slower in humans and other eukaryotes. And it's a complicated process. There's at least a dozen enzymes that take part in all of this process. So how does it actually begin? Well, in order to start this, there's got to be a beginning, all right? Every journey starts with the first step. And we call this point on a DNA strand the origin of replication, all right? The origin of replication. Now, in a bacterial chromosome, remember it's a circular thing, and the origin of replication is at one point in the circle. And the two strands of DNA that are intertwined will begin to separate from there. And eventually, they end up with or forming two completely new DNA strands. We'll talk about this in more detail in a second, but you can see here the origin of replication is beginning to separate out. The same kind of thing occurs in eukaryotic cells like human DNA, except that there are multiple origins of replication, hundreds or even thousands of them. Okay, the replication process starts at one point, but goes both ways in both directions, and you can see that indicated here until eventually you form the two new daughter DNA molecules that are, again, semi-conservative in the process. All right, I'm going to post this PowerPoint on or in the portal, as you know. So please make sure that you go back and have a look at this animation showing the origins of replication. Okay, move myself again. So at the end of each replication bubble, is a fork. All right, so the replication fork is right here. All right, that's what we're looking at here. It's a Y-shaped region where the DNA strands are elongating. Now, there's a couple of things that you need to be aware of here. First, a helicase molecule, which is this one right here, the green, are enzymes that are untwisting the double helix. They're literally, it almost like works like a zipper. It's the molecule that literally unzips the DNA. But the zipper becomes unstable, right? Because this is a very flimsy molecule. And so there are some proteins that get binded to it. These are binding proteins that hold the DNA, kind of stiffen it up a little bit and make the process easier. Then there's an enzyme called topoisomerase, which works at the other end in the still zipped region of DNA that gives it a little bit of stability. What it does is it breaks some bonds and then puts them back together to relieve some of the stress and strain that exists in that molecule. You don't want it breaking apart before you can unzip it. And so that's what the topoisomerase does. So helicase, topoisomerase, and the single-stranded binding proteins. The last thing I want to point out here is that there's another enzyme, a third enzyme called primase, all right, which is building an RNA primer. We'll get to that in just a second. Well, all right, we'll get to it now. So the DNA polymerase requires a primer to which they can add nucleotides. And so the initial nucleotide strand is actually RNA. It's a weird little thing. And we call this little short segment of RNA a primer. And that's made by the primase that you see in the up here in this previous slide. Primase can start an RNA chain from scratch, and then it adds RNA nucleotides. All right, remember, they're the same as DNA nucleotides, except that thymine is replaced with a uracil. And it replaces them one at a time using the parental DNA as a template. The primer is a very short piece. It's only 5 to 10 nucleotides long. And again, it starts at the 3' end. We always go 3' to 5'. Okay, always the 3' to 5'. Then molecules called DNA polymerases will begin to synthesize new DNA molecules at the replication fork. Now, DNA polymerases require the primer. That's why you need the RNA primase, and a DNA template strand, which comes from the original parental chromosome. And as I said earlier, this goes really, really quickly, 500 nucleotides per second, or in bacteria, and what does it say, 50 in humans. So each of these nucleotides that's added to the new DNA strand is a nucleoside triphosphate, 3 phosphates, not just the one that we talked about yesterday. There's a oopsie daisy, molecule called DATP gives an adenine to DNA, and it's similar to the ATP that you see in metabolic activities. The difference is in their sugars. DATP has a deoxyribose sugar and ATP has a ribose sugar. And as each of these nucleotides joins the new DNA strand, it works during a dehydration synthesis reaction. So water is being removed from these molecules. But during that process, it gives up 2 of the 3 phosphate molecules that are there. All right, and those 2 are lost as a molecule called pyrophosphate. Okay, gentlemen, that brings us to the end of the first slide show here for today. Please stay tuned and log into the next one and see where the adventure takes us.